In PyMol, as I told you, there are some ways that how you can open the file. You can open the file by going to File and Open. Another ways by which you can open the file is that, let me lower down this actually, the Zoom screen, which is on the head of mine. Okay, here. Okay, one way was to open the file from here. Other options are get PDB. So when you go to get PDB and your internet is connected to your laptop is connected to internet, for example, I will say 1HRV. If protein structure of this PDB code is available and you say download, then it will be automatically downloaded by connecting to the PDB website directly. I mean, instead of going to PDB website and downloading the things, what you can do, let me repeat it. You will go to File, Get PDB, and if you know the code, for example, I know the code 1HRV, I will type it down and I will say download. It will automatically download the file from the internet. Another way of doing it, let me again clear it up. Another way of doing this is, let me make it like this for me. Okay, you write fetch here. You write fetch and you will ask it 1HRV and you press enter. And it will automatically download the PDB file from the internet. So we got this PDB file actually from the internet, and this is 1HRV, which is basically the HIV surface protein, which I have chosen to give you some demonstration that how the proteins and ligands, they interact with each other. And here in this thing, you can see here is a ligand molecule which is present. And if you look at the sequence, we can see here is only one chain is present. Okay. And there is something is present in the D chain. We actually, if you want to delete that D chain, you can delete it by selecting the residues, means you will click here, and then by clicking shift, you will press here, it will be selected. One way is doing is this thing, and then you will right click on it and you will remove it. So we will have just only one chain and this ligand, which is particularly present here. So we got this analysis. So how we can analyze the thing? I have given you, I have just taught you last time these particular options in which there is action button, show, hide, and label buttons. I will go to action and I will go here in this particular era, and in the preset, I will ask it, give me the ligands. When I will click at the ligands, it will automatically go here and it will show me the ligand molecule. And it will also show me the amino acid to which it is interacting. So here you can see. And now next thing what I can do, I will go to hide and I will ask it, okay, remove the ribbons. So it will remove all the ribbons around, and the amino acid which are just interacting with my particular ligand molecules, they have been shown here. So this is the amino acids which are interacting my ligand molecules. Now what you can do, here you can see, if I make it bigger, here you can see there are two amino acids like this one and this one. These two amino acids, actually, these are forming the hydrogen bonds. So these amino acids are selected here, actually. I will go to action and I will just rename this selection as polar and I will press enter. So this is my polar selection actually. If I want to label them, I will go to L, I will label them. So it has labeled them as a serine 126 and tyrosine 128. And then The other amino acid, I can select them very quickly one by one. So these are the amino acids which are actually forming either the interactions by the van der Waals forces, by electrostatic interactions, by hydrophobic interactions, or non-polar contacts. So these amino acids, they are forming the non-polar contacts with our ligand molecule. So I have selected them one by one. Here you can see that these amino acids have been selected. Here is one left. So I got all of them. I will very carefully check them. So I select all of them. Another way is selecting them. I will show you later that even by using some commands, you can just select these amino acid. And then, so this is now our new, this is our new selection. I will go to action button. I'll rename it to non-polar, for example, and I will press enter. So I got one selection which is polar. I got second selection which is non-polar. If I label it, label of these amino acids will be shown. So these amino acids, these are surrounding our ligand molecule, because ligand molecule is very big actually, and that's why there are a lot of amino acids which are interacting with that. Okay, then I will select ligand. In selection, I will name it as ligand, and I will press enter. So I got these particular things here. So the polar amino acid, for example, I want this polar amino acid to be color as red. I want these non-polar amino acid actually color as blue, for example. So you can recognize them very easily that which amino acid is polar and which is non-polar. So let me color them red. Okay, these two amino acids, they are colored by red and these are blue. And then this is my ligand. I will also color it with magenta. So this is one way of doing that. Another thing, which if you wanna see the ribbon around these amino acids, what you can do by yourself, you can go to action and you will ask it here, copy to object, rename. Okay, so this is your new protein copy, which is automatically formed here. Then you will ask it show, and you will ask it show cartoon. And here you can see that these amino acids, along with all the cartoon and the ligand molecule, that is visualized here. So this is one way of doing that how you can say that. And even you can also color this protein actually by secondary structure, I will say it. So this is how you can analyze, and this is how you can check that which particular amino acids they are interacting with our ligand molecules. Okay, now what you can do else actually, you can go to S show and you ask it to show the surface. Then what it will show? It will show me the surface of the protein. What does mean by surface? Surface is actually the protein surface which is interacting with the water molecules or the amino acid which are only present on the surface. So I will color it actually in the green form. I have colored it green. And now I have selections here like the polar amino acids. The polar amino acids, as we decided that we will color them into red. So here you can see that our ligand molecules is actually here and it's interacting some amino acids which are colored, and it's just going inside from these cavities. Okay, and then because there were a lot of non-polar amino acids, we have colored them into blue. And here you can see that now the non-polar amino acids, these are visible, polar amino acids are also visible. And then ligand molecule, if we just label it as magenta, so we label it like this. Okay, So this is how we can label the different thing, different amino acid. You can see here that ligand molecule is totally surrounding. This may enter in this from this particular cavity, and then further it is interacting with amino acid, and it has just got inside the protein molecule. So this is how, and if you want that surface shouldn't be that much heavy, you can make it lighter. You can just go in the display, and then you can change the settings of the surface. But I will not go into these details. That I just want you people that you go through these two options, the display and the setting. In settings, you got lot of option. Okay, one thing I can show you about the surface, that if you go to the surface molecule, you have got these things: the color, start, wireframe, the solid, pocket and cavities, and these things, the exterior, solvent, means all these things, these are just in your hands. You can change them. And then also you can find that how you can make the surface actually transparent. So you need to find that particular option, that how you can make this surface transparent, and then your ligand molecule, it will be appear to you. So you can also hide the surface from here if you want that. Your surface can be hide here, and here in the bottom you can see that your ligand molecules and all amino acids have been appeared. You can also show, instead of surface, you can also show the mesh. So mesh can be shown, and in mesh you can see that your ligand molecule and its location and the amino acids which are interacting with them. So it depends that what you want to do actually. You want to do actually just only the docking thing, or you want to study the mechanism of the protein, how it's interacting, which amino acids, and from where the proteins they are interacting with each other. So here are a lot of things which you can do by using this, you can say, the PyMol. And also in the presets, you will find many other things. Like if you want to make the pictures which are publication ready and which only consist of proteins, you will go to presets and publication. And here you can see that you will find a publication ready figures actually. So this is your publication ready figure, which you can just save in form of the image by going to the ray, and then you ask it, okay, rate. You can, if your laptop is good, you can go to higher resolution, higher DPI, and then you can save the file actually anywhere where you want by giving it any name, and then you can save it. So this is how you can save save particularly the things which are just similar to the publications or the journal figures. You can also put them into your thesis, and you can study that. The last time I have shown you one thing, that you will go to presets and ligand sites. And in ligand sites, there are certain options like the cartoon thing, and then it will give you figure something like this. Okay. And here from you also give it an analysis, but it's bit difficult because you need to hide the ribbon, and there is a problem in selecting the amino acids which are surrounding that. But this way you can separate the polars and the non-polar amino acids. You can get the surface figures actually very easily. And in presets you have many other options, that how you can make these figures actually. And then you can get the pretty figures, and pretty with the solvent figures, and the protein interfaces actually, where the two proteins, two subunits, they are just interacting with each other. So this is how you can analyze the poses, and you can get the figures which are actually the publication ready. But this all comes after the docking. Another thing is that even if you people are a bit expert afterwards doing the docking in Autodoc, and then you know that what thing is right and what is wrong, and how your programs are behaving, then even you can install the Autodoc plugin in PyMol, and you can directly do the docking by using the Autodoc in the PyMol. Means instead of using the ADT, you can do the docking here. But again, it depends on you, that what you want. Is it the simple docking or the complex docking or any other thing? So this was about the pi mole, and pi moles actually we give its introduction in a very detail, and there are a lot of option else, but I will not go into the detail. If you want to make your videos or extra, then you can also make the movies and the videos by using these spy moles, but I just leave it to you people because that is beyond our scope actually. If you have any problem in that, then you can ask me, and then I can help you with that, at how you can even make the movies in the PyMol. Okay, and here are certain plugin options which I was telling you, that in the plugin manager you get lot of plugin actually. You can align the different proteins, and this part we will do in when we will doing the bioinformatics specialization. And I'm thinking that in the 6th week actually we will start the specialization things, and we will may have the 2 groups or 3 groups. One will be totally for the bioinformatics one, and other will be the computational chemistry ones. In bioinformatics, you will do the things like the BLAST and phylogenetic tree analysis, and how you can analyze the proteins, and how further you can do the MD simulations. While the computational chemistry, they will go into the toxicity analysis and then TFTs, and how you can optimize the ligand using the certain basis sets, and then how you can analyze the results actually. So this will be two different prongs, and you need to select one of them there. So here you can install the different plugins, and if you want to play with these plugins, there are a lot of options, a lot of plugins available online. You can download them, you can install them. I have installed a few plugins on my Mac actually, but not here on the Windows. I will show you someday that how you can actually just choose the your particular plugin, and then you can just download it. Otherwise, you can just put the address here, and you can fetch the plugin directly online. And these are the repositories which actually provide you the certain plugins. If you select any repository, a number of the plugins, they will appear here. And from those plugin, you can you can just check and click that, okay, this plugin I require, this plugin I don't require. So I think I have accidentally clicked on that. So it's downloading everything which is related to plugin here. So it usually takes some time to plug in the things, to download the things. Okay, and if you will know actually the Python, then you can run Python files directly in PyMol, because the PyMol, this is a command prompt of the PyMol, and then you can directly run the PyMol uh, under the Python environment. You just write down the certain scripts, and then PyMol will help you to just get into those scripts. But that is for the advanced thing. Maybe someday or after this internship, we will do some session of the PyMol, which will be for certain advanced users, that how we can implement the certain scripts in the PyMol and write down and make all these figures by using the Python scripts into few clicks, instead of just doing this all efforts. So this is also possible, but you need to be fluent in certain commands, because you need to customize them every time when you are using different proteins and different things. So actually, let me just stop here about the PyMol, because there are a lot of things, and we will continue with PyMol, but I will just stop it. So PyMol in the start, it looked like this, and I hope that many of you have used the PyMol actually. I don't think so. Any other thing which is uh, necessary to you. I have told you about almost everything which you are gonna do in the analysis of the docking. So I will just, one thing is important, that sometimes when you open the protein, then you can analyze that protein. For example, I will open this protein, which previously we used. We need to check them, the gaps, that certain amino acids are missing. Again, here is a gap. Again, this protein is starting from 4. Some proteins, they start from 11, 12, 13, 50 even. So we need to check that what are the gaps and why these gaps are present. Because sometimes the scientists who have done the X-ray crystallography, these particular amino acids, they are present in the regions which cannot be forming the structure, or they cannot be solved by using the NMR or X-ray crystallography. So they just leave it as such because they don't know that what kind of structure will be present here. So one option is that just leave it as such. But if it's interacting with your ligand molecules, then you have to model it by using the homology modeling, as I told you in the very start, or you just need to get the protein from the another repository like that for code. I will show you that, all of you, I hope so. So that how we can model the proteins actually in the start if our receptor is not present in the database, in the protein database. So that is a big question, that what we will do if our target protein or receptor is not present in the structural database, in protein data bank, then what we will do? Of course, we will do either the homology modeling or the ab initio modeling, or we will get it from some other database which is based on the theoretical models, not the experimental ones. Okay, let me just finish it here. And then I want to show you the Autodoc Vina thing, that how we can prepare. So one way of doing the Autodoc Vina is actually, let me open the Autodoc tool and then I will share its screen. So Autodoc tool, let me open it. I will go to Autodoc tool 1.5.7 and let me share the screen. So we are somewhere. So it's just, I think it's just opening. So it's running the Autodoc tool from this command prompt, and then it will open its graphic user interface. So MGL tool is basically the graphic user interface of Autodoc. There are many graphic user interface of Autodoc. One is this MGL tools, other one is Pyrex, which is very famous. And then there are other like the Discovery Studio and then Flex and other things which also are based on. So I think only one window is visible to you people. So why not I share the appropriate one, which is this one. So now Autodoc tool is actually shared. So usually the people, they run the Autodoc Vina from here. If you run the Autodoc Vina from here, first you need to download and install the Autodoc Vina. The Autodoc Vina, from where you will install, let me share the screen, I think altogether where it is. Okay, it's somewhere here. So I have just opened the windows for you people, that where you can download the Autodoc Vina. It's somewhere on the scroll. Yeah, this one. So this is the place from where you download the Autodoc Vina for Windows. So you will go and you will download the Autodoc Vina, and then it will be downloaded for you people. You will ask it to download and it will be downloaded in few seconds. So this is how the Autodoc Vina is actually installed. It's downloaded. So it's downloading. Meanwhile, I have downloaded it somewhere. Why not I open it from there? Somewhere I have put that. Okay, maybe here. I have just put the Vina. So Autodoc Vina will be automatically installed actually on your laptops. I will send you the Autodoc Vina tutorials I have made for you people, so you can just follow the Autodoc tutorials. So this is core fetch. So here is some time there is an error. What kind of error? Let me show you, because this is the reason that I have just shown you that how you can download it instead of giving you name. Actually, when you first time download it, it download the things in form of math libraries or something. This is not actually the file which you are looking for. You need to check only the Autodoc Vina file actually. If you have gone towards the wrong link, it will not open. It will open something which is some kind of math libraries and libc, libm, something like that. You don't need that actually, because I just found it yesterday when I was downloading this program for you people, that this source forge actually, it has got some errors sometime. So in Autodoc Vina background, it also got some error. So here you can see that you have to go to files, and then here are certain files, and then you have to download from here. And then downloading from here, it will download the files. And even if you want to download the previous versions, you can also go to the files and download the previous versions which are related to the Mac and the other things. So I will also send you actually the file which is a stable one. I got the Vina file which is 1.2.3 actually. And that is very stable version of the Vina. And I will send you that one. I will put it into some link and you can use this file directly actually. And this is very stable version of Autodoc Vina 1.21. Okay. And other thing from where you can get running the Vina, I prefer that I run the Vina on the command prompt. I will show you that how we can run it on the command prompt, means in the Ubuntu. And Ubuntu is working very best for me when you are running the Autodoc Vina. So let's see. I will, I think, restart it because I don't want it that we just stop in the middle. So I need you people to download the Autodoc Vina. If you are unable to download from internet, I will send you the stable version because this is getting some errors. I don't know why, but it's there. This source forge is getting some errors in downloading the things. And I have these Jupyter libraries will do it later on. I hope so in the 5th week. I have made some Jupyter notebooks for you people on which you can do the online docking actually, instead of using these things. Even on your cell phone, you can do the analysis of the files, and I will show you that how even you can do that. So let me just restart it, and then we will do something about the Vina properly, and we will look at that, at how the Vina works. Let me start and rejoin, please. Okay. Or we can just have a gap of 10 minutes maybe for the prayer, and then we will be back at 6:17, 6:18. I hope so. 6:18 we will be back. I hope so. Okay.